The overall goal of this procedure is to study brain tumor-initiating cells in vitro. This is accomplished by first mechanically and enzymatically dissociating primary human tumors and then culturing them in stem cell serum-free culture conditions. Next, sphere formation is monitored over 3 to 5 days. Tumor spheres are then enzymatically digested again, and their surface markers are evaluated by flow cytometry. The final step is to sort the desired cells into 96-well plates or ampligrid slides for further characterization. Ultimately, the self-renewal ability and differential gene expression of the sorted population can be assessed by limiting dilution analysis and single-cell RT-PCR.
The main advantage of this technique over existing methods, such as bulk tumor analysis, is that it prospectively isolates the brain tumor-initiating cell or cancer stem cell populations. We first had the idea for this method when we applied the culture conditions originally used for normal neural stem cells to a variety of human brain tumors, and found that this culture method specifically selected for stem cell-like populations. Demonstrating the procedure will be postdoctoral research associate Chitra Venugopal, graduate students Branavan Manoranjan and Sara Nolte, and our flow cytometry technician Nicole McFarlane, all from our laboratory.
Begin by adding 200 microliters of thawed liberase to 15 milliliters of artificial cerebrospinal fluid, or ACSF, and then placing the tube into a 37 degrees Celsius water bath. Bring ammonium chloride solution to room temperature at this time as well. Then, add 5 milliliters of ACSF to a specimen container of brain tumor tissue, swirl the container to rinse, and then pipette off the fluid to remove the red blood cells.